Hello, and welcome back. So in the last few videos, we were working with an example, an independent samples t-test or two sample t-test regarding some data that Charles Darwin, old Chuck, gave us, okay? And just as a little recap, let me kind of go back here. Nice. So we were looking at the difference in heights between cross-fertilized plants and self-fertilized plants, a specific type of pea plant or something. And we wanted to see, is there a difference between these two samples? And then can we blow up what we found in this sample to the whole population from which these two things came? Okay. And it turns out that, yes, there is a statistically significant difference between these two types of plants, these two fertilization methods here, because we found a p-value. We got a p-value of less than 0.05. Okay, so in this case, there is a 2.357% chance that the difference we saw was due to just total randomness. Okay. Now, we were then able to say, well, if we were to blow that up to the whole population, to every single pea plant that cross-fertilizes and every single pea plant that self-fertilizes, we'll find that on average, there's going to be a difference between those two groups of at least 3.8 inches to at most 4.82 inches. Okay. What the 95% confidence interval tells us is that if we were to go out and resample, get similar size samples each time, but with different data, 95% of the time, we'll find that difference to be in between these two values. And then 5% of the time, there'll be something different. And that's pretty good. You know, that's 95% of the time is pretty frequent, at least for us in statistics. Oh, boy. Okay, so the deal here is that we have two independent groups, okay? These two groups, it's not like we have a pre and a post sort of assessment or anything like that. We just have two independent groups. We were able to measure these two things essentially at the same time, okay? What, though, what if we just have paired data? Or not just have, but what if we do have paired data, okay? And that is to say that we have sort of something like this example here, where we have the same people in the pre-intervention group as in the post-intervention group, okay? So it's the same people or same plots of land or same plants or whatever it may be, but we have this kind of component of time, a pre-intervention and a post-intervention, or pre-experiment and post-experiment. Okay, and you're probably pretty familiar with this. A lot of drug trials are like this. A lot of exercise testing is like this. After giving some people, some participants, an exercise protocol to follow, you test their fitness beforehand, test it after they do the exercise protocol, and see how it changes, right? So we have this example here. We've got to do a slightly different test. Thankfully, we have a few ways we can do this. Also, the code literally takes 2 seconds again to run. So let's get into it after all of that. So the FAA wants to figure out if it's okay for airline pilots to get absolutely blitzed before flying a jet with hundreds of people on it. They know that we have a good rapport as far as the type of analysis we can do, and they want to give us a 100K. So a ton of money. We're already pretty rich, so we don't need that money, but we're going to go ahead and say yes. Okay. So what these data contain are measurements of reaction time in milliseconds before and after consuming alcohol. Okay. And we've got 10 of the same pilots. Okay. So these two people, for instance, you tested their reaction time before consuming alcohol, and then you tested it again after they had 2 martinis in the cockpit. Okay. Now, of course, I'm not condoning drinking alcohol. You can do whatever you want so long you do it safely. And of course, I'm not condoning, operating big mechanics after drinking alcohol, especially flying a jet. Okay. So why do I have one sample and paired t-tests up here? Well, if we go back, way back, way back, we actually have a bunch of different types of t-tests we can do. In this case, with the independent samples t-test, we got 2 different groups, okay? But it turns out that if we have paired data, we can actually do a paired t-test or a one sample t-test. This in R, the paired t-test, is the easiest to do It's the simplest code. This takes a little bit more programming to do, but it literally is just some arithmetic. Okay. Again, the computer does it for us. So I'll show you how to do both, and you can kind of pick your favorite, because it turns out that these two things give you the same answer. Whoa. Okay. Now, before I give away any more spoilers, let's kind of talk about that. So a lot of the time, sometimes we want to create a new column of data based on the other two. Okay. So if we wanted to go ahead and calculate the difference between these two columns, we could just say, take this column and subtract this column and put it in this column. It's as easy as that. And we have some code to do that using the tidyverse. You can ignore the dplyr : this is the function here that we're going to be using. And we can kind of see the differences between these two. We are going to be using the mutate function, not the transmutate. Whatever, no biggie. Okay, so let me go to R and kind of show you what I'm working with. Now, I've already got the code here, and you should have this code as well. You can run it along with me. But I've got my working directory set. I've got all of my data. We're going to see that this list here of stuff is going to get very long as the semester goes on. But let me just go ahead and read in the data. And already we'll see here that we've got something kind of funky happening. We've got 15 observations, even though the data said, or at least the PowerPoint said we should have only 10. Well, what's the deal? Well, if I click on the data and it brings it up, something weird happened with the storage of these data here. We've got a bunch of missing values, not available values from rows 11 through 15. We also have this whole column here of missing values. Again, I didn't do this intentionally. This is just what happens sometimes with CSV files. You'll find this in real life. You'll find this, pretty frequently. That is okay, because if you recall, we can use the square bracket notation to select the columns the rows that we want to keep and the columns that we don't. Okay, and so I've kind of got that translation here, where I want to keep rows 1 through 10, and I want to get rid of column 3. I want to keep these rows here, and I want to get rid of this third column. Okay. And if I just run this, I'm putting it into the same named data because I don't want an extra thing running around here. I just want to kind of override what's already there. So if I run that, we'll just check it out real quick. Booyah. Look, it's real nice and clean. Super nice and clean. Now, the next thing I'm about to do is not totally necessary for you to know exactly what's going on here. But since these data are paired, I actually want to have another column here of the ID number, essentially, because we know that this first row right here is one pilot, pre-boozing it up and then post-boozing it up. Okay, and then this is another pilot, and then the same pilot again, and then a different pilot, and then that same pilot, so on and so forth, right? And I kind of just want a column here to denote the fact that these two kind of cells here, this whole row here, is actually one person, okay? And so I'm going to use this code here on line 8 to do that. And let me kind of break this down briefly, just so you can kind of see what's going on. If I highlight this 1 through n row of reaction, okay? Well, if I actually just highlight n row, this is just going to supply me a value, and it's going to tell me the maximum rows that are in our data, or the total number of rows, and it's 10, okay? And if I do 1 colon that, effectively that's just telling me, or telling the computer, count rows 1 through 10, and it's going to give me 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, okay? And then all I'm doing is I'm saying, well, these 1 through 10, these numbers here don't actually mean anything numeric. These are actually representing people. So I could have typed in, Bob, Susie, George, Elaine, Jerry, Kramer, Newman, right here, and it would represent the same thing. So when I say this as.factor, I'm telling the computer to say, hey, don't treat these numbers as numbers, treat them as essentially names, okay? And then what I'm doing here is, well, we don't have a column right now called ID, so I'm going to make one. And I just use this $sign here to say, this is totally empty. This does not exist. In fact, if I run this, it's going to say null. So it's totally empty. I'm going to supply it this information, and then we'll be good. Okay, so if I run this, check it out. So I've got ID. I could have called this thing here anything. I could have called it participant, could have called it whatever, group. But I called it ID just to kind of remind me what these rows represent. And then I've got, IDs 1 through 10, pilots 1 through 10. Okay. Oh, boy. And then the next thing I want to do is I always want to read in the tidyverse because we've got to visualize these differences here. I want to make a box plot. That's kind of always a good idea. Ouch. This is my knuckles. I want to visualize these differences, which is always a good idea to plot a box plot every time you do a t-test. Okay. And so the way the data is right now is that I want some categorical column here. I want some column denoting the fact that we've got a pre and a post, okay? Because I want that to be on my x-axis. But right now we don't have anything here that will give me a box plot with categorical labels, we'll say, on the x-axis. So I have to do some manipulation here. And we're used to seeing this, or at least we have seen this before, where we gather the data and essentially just stack it on top of each other. So if I go like this, well, let me kind of go and tell you what each one is just real quick. Intervention, I'm just going to say that that's going to be the column of pre and post. Reaction time is going to be obviously these numbers here, okay, stacked on top of each other. And I just want to grab pre and post, okay? I don't want to grab the ID column. I just want to stack these two on top of each other. And then factor key, that's just going to make sure that the column names are treated as factors instead of characters. So if I run that, go ahead and look, check it out. So I've got the ID here for each one, and we can kind of see how the stacking works again. So I've got first pilot here, and then 10 more down, I've got the first pilot there again, just denoting that it is the post assessment. So they're properly, oh gosh, they're boozed up at this point, okay? So first pilot, second pilot, pre, second pilot, post, right? And then we have the reaction time here. And then we've seen box plots before, so I'm just going to run this. You can kind of see what I'm doing. Let me move this down just so you can see. Very cool. All right. Okay. So what does this box plot show us? Well, with reaction times, this is kind of like, I don't know if you've ever done this before, but maybe in a biology lab or a physiology lab or something, where you hold a ruler, somebody holds a ruler right in between your hand, and then they drop it kind of randomly and you have to grab it. And then the distance that it falls, you measure, and then that's your reaction time, whatever. That's essentially what they did here, okay? I'm sure it was a little bit more complicated than that, but that's the general idea. So the smaller times here, smaller reaction times, means that they reacted faster. And anything longer, well, took them longer to grab the ruler or whatever. Okay, so what does this show us? Well, it kind of shows us what we thought, or at least what we know, if we know anything about booze, is that when you're totally sober, your reaction time is, decently fast, or it just is what it is. But when you drink, your reaction time slows down quite a bit, okay? And that's kind of represented here. However, what this does not show is the paired nature. This is kind of treating these two groups as two independent groups, or at least this is what the box plot is showing, okay? It's not taking into account the fact that we've got the same people pre and post. This is a kind of just visualizing as if they were totally independent from each other. And I want to make a plot that shows that paired nature. And I kind of want to see how each individual changes from pre to post. Now I've got this plot, this code right here already up and running. You do not have to know how to do this. The plot I'm going to show you, to be totally honest, I think we'll ever only see this plot this time. I don't think we're ever going to do anything in the future with this type of plot. However, it is good to know. Good code to have just in case you want it for a homework or something like that. So if I run this, whatever, we're not going to so much worry about what's going on here. We've got a nice little plot here, and I'm going to zoom it in just so we can take a good look, and then I'll stop the video and we'll do the proper t-test. So what is happening here? What is this showing? Well, this is showing each individual pilot here, pre and post booze, okay? Reaction times pre and post booze. I've got it up there. And we can kind of see what the box plot showed us. However, we do have now more information. We got a little bit more granular with our looking glass, we'll say, okay? So on average, it looks like most people have increasing reaction times after they drink, okay? So like this pilot, you know, we have kind of ID over here. This pilot 3 here was really affected by alcohol. Their reaction time really increased after drinking. But you have people down here who were pretty fast to begin with and stayed pretty fast. They still increased, but it's taking into account the fact that they were already quick to kind of begin with, okay? We also see some interesting things here with these two pilots, okay? These two pilots were, you know, kind of on the upper end of kind of baseline reaction time, but they actually went down. Their reaction time improved after drinking. And we want to account for this, okay? We want to account for these kind of things. And that's what that paired t-test is going to do for us. And then the last kind of thing here is that we have somebody who was really, really slow. Their reaction time was almost a second long, unchanged after booze. So they stayed slow after drinking. They were always slow, and then their status quo was still the status quo for them. They stayed the way they were. But we want to account for these kind of things, okay? If we didn't want to account for these kind of things, we would just sample 2 independent groups, okay? But we know that people are going to behave slightly differently from each other, and we see that here. So we've got really fast people staying fast, and we've got really slow people staying slow, and we have some people who are greatly affected by the intervention. In this case, drinking a martini, okay? So this kind of behavior is what a paired t-test is going to account for. And also, since we have paired data, since we have the same people pre and post, we have to do a paired t-test or a one sample t-test, which I'll show you in just a second, okay? So with that little intro into the data, a little intro into why we want to do a paired t-test, I'll stop the video and we will pick it up here momentarily. Okay.